And as you can see from this picture, Galicia landscape is dominated by mussel wraps, which are these.
This is the one. This is that one. There's a little video. It's a video. If you go to that.
But I want to go back though.
Oh, you want to go back?
Yeah.
Okay.
Which are the floating structures you can see. Those are called rafts, and they're used for the culture of Mytilus galloprovincialis, which is the native mussel that lives in the Galician Rias, in the northwest of Spain. So now before we start, let's take a little splash and see how the mussel farms actually look like underwater. So what you can see in the video are the ropes used for the culture of Mytilus galloprovincialis. There's more than 3,000 rafts in the Galician Rias. The ropes are 12 meters long, and there's 800 mussels per meter of the rope, so a lot of mussels go together. And mussels clear between 3 and 5 liters of water per hour. They get all the particles suspended in the water removed. So Xenostrobus securis is a mussel native to the coasts of Australia and New Zealand. And it was most likely brought by ballast water of ships or shellfish farming activities to different countries like China, Japan, Italy, and Spain. So nowadays, Xenostrobus securis, this invasive mussel, can be found in Galicia, in this area that I show you in the video. And it's probable that the successful colonization of Xenostrobus securis was sustained by its tolerance to extreme salinity variations, because it can be between 1 to 35 parts per thousand of salinity, although high mortalities occur above 35 parts per thousand. So in Galicia, you can see in the map where Galicia is located, in the northwest. This alien mussel coexists with the native mussel that I show you in the video. And it can only be found in the intertidal of the innermost area of the Ria de Vigo, where the salinity is around 28 parts per thousand. The engineering activities of non-indigenous bivalves like Xenostrobus securis can significantly modify the ecosystem structure and also cause important economic losses. By engineering activities, I mean the physical modification of the environment through the filter feeding activities, the shell production, and also the biodeposits, which are the feces and the pseudofeces produced by the bivalve. There is 250,000 tons per year of mussel Mytilus galloprovincialis produced in the Galician Rias. And it is unknown whether this alien species could attach to the suspended ropes. In addition, the exotic mussel could compete for food and space with the native filter feeders and reduce the population of native mussels. As you're going to see in the next slide, this could decrease the natural availability of Mytilus galloprovincialis seed, because in Galicia the mussel farming relies on seed obtained either from artificial collectors placed in the rafts or seed that lives in the intertidal area. Then once we obtain the seed, we place it around ropes, and it grows for 5 to 6 months until 50 millimeters shell length. Then mussels are detached and to constitute new ropes with less density. And lastly, we let them grow for 9 to 10 months until they reach 70 millimeters shell length. So in this study, we aim to compare the annual biochemical composition and the physiological energetics of both species to to increase our knowledge of the relationships between the invasive mussel, Xenostrobus securis, and the native species, Mytilus galloprovincialis. What you can see in this picture is the innermost area of the Ria de Vigo, where the study was conducted. So to reach this objective, we sampled both species from the mixed mussel beds that colonize the rocks in the innermost Ria de Vigo. This was hard work because we went there every month and we measured the environmental conditions. This is the salinity, temperature, chlorophyll, particulate matter. And then we also took samples from both mussel species to measure the protein, carbohydrate, and lipid content of the mantle and the digestive gland. In addition, we performed in situ physiological measurements that included the ingestion rate of both mussel species, the absorption efficiency of the food in the mussel's gut, and the oxygen consumed by both species. Our results revealed that Xenostrobus, the alien species, had comparatively higher proteins and glycogen in the mantle and the digestive gland. So we think that proteins could fuel greater tissue and cell growth, while glycogen could provide energy for growth during low tide. This is when there's no oxygen, or energy for reproduction after being converted to lipids. Because unlike proteins and lipids, glycogen can be converted to energy without activating the Krebs cycle. However, the native species, Mytilus galloprovincialis, accumulated more lipids in the digestive gland and also show a higher ingestion rate than the invasive species, as you can see in the graph. We observed that in mixed mussel beds, the native mussel always colonizes the top of the aggregations. And this distribution could explain the higher ingestion rate and the lipid levels, because it's probable that the native mussel has an easier access to the lipid-rich phytoplankton or microphytobenthos compared to the invasive species. In addition, the digestive gland is the food processing organ, so it always reflects the composition of the diet closer than any other tissue, like the mantle tissue. It is well known that lipids provide the densest form of energy in the marine environment. And the higher amount of lipids found in the digestive gland of the native species seem to compensate for the lower levels of carbohydrates and proteins relative to that of the invasive species. This was demonstrated by the similar energy content found both in the mantle and the digestive gland of both mussels. So in conclusion, we think that the higher accumulation of proteins and glycogen in the tissues of the invasive species compared to that of the native mussel suggests that this is a species that is well adapted to live in the intertidal shores of the innermost area of Ria de Vigo. However, the inability to tolerate salinities higher than 35 parts per thousand could restrict the further expansion of this invasive species to the outer parts of the environment. So thank you very much.